Hi, I'm going to focus today on emerging targets for adnexal high-grade serous cancers with a focus on PARP. This is an exciting area. It's a field that's finally starting to come of age, I think, and there are a lot of exciting new areas. Many solid tumor targeted therapy talks start with a slide that looks like this and identifies some really horrible activated pathways and ends up talking about some novel kinase or monoclonal that has remarkable activity and minimal toxicity. We have not had such luck in the last couple of decades with ovarian cancer. Most targeted therapies that have been successful in other cancers have fallen flat in ovarian cancer, with the exception possibly of the mTOR inhibitors and sorafenib, but their toxicity has precluded their further development in this area. A notable exception, of course, is bevacizumab, which Dr. Berger, I think, will talk more about later. So ovarian cancer is special. It's different, as I'm sure you've all heard today. We're going to focus today on three major areas: tumor suppressors, immune checkpoint blockade, and surface targets as emerging rational targets for ovarian cancer. So as you've heard and will heard, really the molecular abnormalities that are being seen in different subtypes of ovarian cancer probably explain why we're not seeing the same benefits from these other targeted agents. In the rare subtypes of epithelial ovarian cancer, we see a variety of activated oncogenes, but in high-grade serous ovarian, the majority of defects we see are in tumor suppressors. We do see pathway alterations, but sometimes they're activated secondarily from the absence of tumor suppressors. So let's talk first about some of these tumor suppressors, the common ones that are lost or inactivated in ovarian cancer. P53, almost necessary for the diagnosis of high-grade serous. The most important TSG you've never heard of, OPCML, the opiate binding protein, and of course BRCA2 and homologous repair defects in about 50% of patients in the TCGA analysis. So these two, the P53 and the homologous repair, really contribute to genomic instability, which is a major molecular feature of ovarian cancer. We're going to talk first about targeting this feature. So synthetic lethality was first named back in the 1920s by this handsome geneticist who proposed that two genes are synthetically lethal if the absence of either one is compatible with viability, but the absence of both would result in lethality, first shown in animal models in the 40s in Drosophila, and now applied as an approach to cancer therapy. One might argue that induced essentiality, meaning the reliance of a redundant pathway, is more accurate, but we do use the term synthetic lethality. Here we see the pathway of how it applies with BRCA inhibitors. PARP inhibitors. Typically, we get single-strand breaks that are repaired by base excision repair, which depend on PARP. When this doesn't happen, we're more likely to acquire double-strand breaks. In the absence of homologous repair recombination, we get cell death. This is the basis of the development of the PARP inhibitors. As you'll see, many companies have jumped in the game. Of note, Olaparib, which there was excitement about, might not actually inhibit PARP, but the remainder of the drugs are in the field. We're going to talk first about Olaparib, which was really the first PARP inhibitor to show meaningful clinical activity. The New England Journal phase 1 dose escalation study by Fong, which looked at 60 patients with breast, ovarian, prostate cancer, about a third of whom had germline BRCA mutations. There were some impressive responses, notably in the BRCA mutated patients, prompting further development. So let's review once again. Single-strand breaks if you inhibit PARP, double-strand breaks, and we would expect that in homologous deficient cells, we would get lethality, and those who have functional homologous repair, we would not have an effect. Indeed, when Olaparib was given to patients with and without germline BRCA mutations, we do see response rates in those with germline mutations, but we also see less but some response rates in those without. So there's obviously more to the story than the simple model. Auday showed that there's a dose responsiveness, and 400 milligrams POBID is really the standard dose used now for Olaparib, which is more effective than the 100. Another seminal paper with Olaparib was by Lethality, published in the New England Journal, showing in patients who had responded, who had platinum-sensitive recurrent disease who had received a platinum doublet, patients were then randomized to receive olaparib as maintenance versus placebo as maintenance. In this trial, there was a clear PFS advantage in the overall population, which include germline BRCA mutants and those without germline BRCA mutations. And in a subset analysis, those with germline BRCA mutations appeared to derive a survival benefit. And based on this, the European medical regulatory agencies approved olaparib for this indication, but the FDA did not. It was really this study that served as the registration trial for olaparib in the United States, a non-comparative analysis involving multiple tumor types of patients with germline BRCA mutations, where an impressive response rate was seen in ovarian cancer, as well as a 7-month median progression-free survival, leading to FDA approval in December of last year. Of note, olaparib was active in pancreas and prostate cancer and probably will be developed in those cancers as well in upcoming trials. So let's go back to this area that we noticed before, that some patients without germline mutations still did have impressive responses to olaparib in some of the earlier trials by Gelman. What is this all about? And it seems that the 50% of patients who have some sort of homologous repair defect may hold the answer to this. So in the TCGA, homologous repair, again, in 50%, some of those were explained by germline BRCA mutations, others by acquired somatic BRCA mutations, and 20% other. And as we've heard, there's been some elucidation of what the various mutations are that contribute to this, and they have been variously described in terms of BRCA-ness, homologous repair defect signatures, or more recently as genomic scarring. Importantly, rucaparib, one of the other PARP inhibitors that's in prominence right now, is using this concept of genomic scarring to prospectively develop a biomarker to basically quantify HRD and identify subpopulations without germline BRCA mutations who might benefit from PARP inhibition. In the Ariel 2 study, in the first part, patients were classified into 3 molecular subgroups: BRCA mutant, BRCA-like, or those who are biomarker negative. This is a graph where genomic loss of heterozygosity was basically quantified as a way to estimate HRD. In the second part of Ariel 2, the signature, the biomarker of LOH, was validated prospectively based on part 1. You can see here some of the genes that were identified to contribute to this BRCA-like LOH phenotype. And as you'll see in this waterfall plot, the ultimate trial showed numerous impressive responses, both in patients with germline BRCA mutations and in those with somatic mutations. Ultimately, overall response rate 82% with an impressive PFS in BRCA mutants, as you might expect. Biomarker negative, similar to the Gelman study, 21%, but this BRCA-like phenotype is enriched in terms of overall response and duration of response, and this may prove interesting for development of this drug as an FDA-approved agent going forward. One thing that we're starting to see is that resistance certainly does develop to these drugs, and the importance of BRCA and PARP to each other and their codependence, I think, is illustrated by this very impressive example of a resistance mechanism. This is a patient with a germline mutation in BRCA2, actually a male breast cancer patient, who initially responded to PARP inhibition and then developed a resistant lesion in his pelvis, which was biopsied and genotyped and found to have a BRCA2 reversion. So no longer loss of heterozygosity, has a functional BRCA2 mutation, probably explaining the resistance. Many questions remain about PARPs. Currently, they are approved for relapsed ovarian cancer that in patients with germline BRCA mutations who have received at least 3 prior cytotoxic chemotherapies. Is there a role for PARPs in upfront treatment? That is under development right now. Maintenance strategies, is that really the best time? And does that make sense in terms of quality of life and cost? Should these drugs be used in platinum-sensitive or resistant patients? They seem more active in platinum-sensitive patients, but so are many other drugs. And what will be the role of the HRD signature and other combinations that are being developed? There appears to be a rationale for combinations with VEGF inhibitors as well as immunotherapy drugs, as we saw at ASCO this year. Looking at another important drug, P53, it's been fairly elusive with regard to rational targeting, as important as it is, it's a hard mutation to fix. There is a drug called AZD1775, which made a splash at ASCO this year. It's a WEE1 kinase inhibitor, which in combination with chemotherapy in ovarian cancer patients who have P53 mutations, pretty much all of them, there was activity in platinum refractory patients, which was fairly impressive. So this is kind of an intimidating diagram, but I'll try to break it down for you. I really think this was an excellent article. If you have interest in the subject, Sabine Mueller wrote this accompanying editorial to the AZD1775 phase 1 data in JCO this summer. Basically, the idea here is that in the absence of P53 as a regulator of the G1S cell cycle transition, the cells are more dependent on G2M halting by CDC2. This is the time where they undergo DNA repair before going ahead. So whether or not CDC2 does this halting is determined by a balance between WEE1 and CHEK1, and the AZD WEE1 kinase inhibitor basically forces the cells to go into premature cell cycle and mitotic catastrophe. That's the idea here. The studies with AZD are now using P53 mutations, absence or gain, very plus minus, as a biomarker for whether to use this inhibitor, but the devil's in the details. There seems to be other factors that may be relevant. Some preclinical models say that P53 loss is required. Others say it does not. In the phase 1 data, loss of BRCA1 seemed to be the strongest predictor of response to WEE1. So this is a drug that I think we're going to hear a lot more from going forward. Multiple questions remain. Are specific molecular subtypes, different types of mutations of P53 important and relevant to this? And it's an extremely complex molecule, and many mutations have different effects on it. And what sort of combinations are rational and usable? And ultimately, does this inhibition actually improve clinical outcomes remains to be unknown. The opioid binding protein, very interesting drug. It does not contribute to genome instability the way the other tumor suppressors do that I had mentioned. It's actually a GPI-anchored protein that floats around on the surface of the cells and can inhibit protein, basically receptors, HER2, FGFR, and FA2, and their pathways. So in the absence, again, 90% of high-grade serous are missing this protein, and these pathways may be activated unhindered. In this interesting preclinical study, recombinant OPCML can actually start to inhibit these pathways. So that has not yet reached clinical use, but is a very intriguing pathway. So let's switch gears here for a moment. Many of you, of course, are familiar with the tremendous benefits that have been seen by immune checkpoint blockade in a variety of other tumors. The priming phase being dependent on MHC-dependent recognition by the T cell and a co-stimulatory molecules, and inhibited by CTLA-4/B7 binding, blocked by drugs like ipilimumab. Ipilimumab and other CTLA-4 inhibitors have not really shown tremendous promise yet as monotherapy in ovarian cancer. However, inhibition of the PD-1, PD-L1 pathway seems to be more interesting and more tolerable based on recent, very new data. In the effector phase, again, MHC-dependent T-cell cytotoxicity can be inhibited by these proteins, but these can be blocked by a variety of drugs. The only published data so far, JCO in September of this year by Hamanishi, reported a small phase 1 trial of nivolumab, a PD-1 inhibitor. This was performed in Japan with 20 patients at 2 doses of nivolumab. 15 patients had high-grade serous ovarian cancer, 2 had clear cell, and 3 endometrioid. There were 2 very impressive complete responses, which were not only clinically dramatic and meaningful, but also persisted in up to a year and ongoing follow-up after completion of a year of nivolumab. Of note, one of these responses was in an ovarian clear cell patient. 2 out of 2 clear cells did respond in this trial. There were some early data reported at ASCO this year with avelumab, which is an anti-PD-L1 monoclonal antibody, which may have some ADCC activity as well. Similar response rates between 10 and 15% were seen with this drug, and multiple patients seem to have stable disease for a prolonged amount of time. That seems to be the major clinical benefit for this class of agents so far. Another monotherapy trial of pembrolizumab, an anti-PD-1 drug, was presented at ASCO. Smaller trial, similar results. Of note, in the avelumab study, 2 out of 2 patients with ovarian clear cell cancer also did have responses. That does seem to be a subtype that is particularly susceptible to these drugs, even though the numbers are quite small. Some questions remain right now. The only published data are in PD-1, PD-L1, and nivolumab. We're eagerly awaiting initial results from the ENERGY-3, where patients with recurrent ovarian cancer receive nivolumab with or without ipilimumab. Which tumors, high-grade serous, appear to respond to some degree, maybe not as dramatic as lung cancer or melanoma, but other factors like BRCA, mutation load, and how many prior treatments might be relevant. Which patients, we still don't know. And a lot remains to be learned and seen, but it's an exciting time. I'll briefly review some surface targets. CA125 was investigated for a long time. There were some monoclonal antibodies against CA125 and also an anti-idiotype antibody that was a vaccine, both of which showed some early promise about 10 to 15 years ago, but large trials with these drugs did not show much toxicity, but were unable to show clinical efficacy. CA125 might also have an immune inhibitory function, so possibly could be relevant in some other contexts going forward. New York ESO1, also a common surface antigen that has served as a target for vaccinations, and immune responses are seen, but has yet to show clinical efficacy in any of these approaches. Folate receptor alpha has received a lot more attention these days because of some interesting drugs that are emerging. Similar to the opioid binding protein, folate receptor alpha is a GPI-anchored protein. It is not a transmembrane protein, and it's frequently internalized. It does bind folic acid, but is probably not the main transporter for metabolic folic acid, and seems to have some other functions which have been poorly described. It might actually serve as a transcription factor itself. The folate receptor alpha is present mostly in embryonic tissues, and it develops to the formation of the neural tube closure. That's why you have to take your folate when you're pregnant. But in adults, it seems to be minimally present, perhaps with the exception of type 2 pneumocytes and a few other areas. So what has been done to target folate receptor? Multiple attempts. There's a monoclonal antibody called farletuzumab, which has shown to be safe, but has yet to show convincing evidence of clinical activity for this drug, despite some ADCC in preclinical models and anti-tumor effect in models as well. A small molecule drug conjugate called vintafolide earned some excitement from some intriguing early phase 2 data in JCO. This is a combination of basically a vinca alkaloid and folic acid, but ultimately this drug failed in phase 3 and is probably not going to be developed further. More interesting is the ADC, the antibody drug conjugate, mirvetuximab soravtansine, otherwise known as IMGN 853, which actually has obtained orphan drug status by the FDA. Let's talk a little bit more about this. It's basically a monoclonal antibody recognizing the folate receptor alpha and has attached to it some mitansine antitubule toxins, which are stable to outside of the cell, but once internalized are enzymatically cleaved and released and exert their influence on the cell, and it can actually go through cell membranes and attack neighboring cells. So early data from the phase 1 dose escalation study, which looked at a variety of solid tumors that were shown to express folate receptor alpha on the surface, showed some impressive clinical and radiologic responses for a dose-finding phase 1 study. There were also several patients who remained on therapy with stable disease for a long time, and a large multi-center phase 2 trial is now planned for this drug, and it should be developed in many ways going forward. That was a very rapid, broad review. I hope you took that all in. I only had 20 minutes, but there was a lot to talk about. Anyway, thank you for listening.